Okay, let's see. There we go. All right. How's it going, everybody? Hope you are enjoying your night. I know that some of you are probably out right now at that Palooza, so I hope you're enjoying that. Otherwise, I hope you're having a good night in. And so let's go ahead. We'll just jump into it, and we'll take off where we last left off. And so, where I remember leaving off is talking about PCR, went over how PCR works, talking about its purpose. And so, if you don't recall, the main purpose of PCR is to make a bunch of DNA copies of whatever sequence we're interested in. That's really all it's for, and it's like a necessary precursor for some experiments downstream. So, for example, if we want to do DNA sequencing, we very often have to amplify the DNA to sufficient amounts first. Um, but sometimes it's used for gene cloning instead, where we want to make copies of the gene within a bacterium, but first we need to make sufficient copies of um, the product, or sorry, the gene using PCR. And so it might sound like a little counterintuitive because you're probably thinking like, I thought gene cloning was to also amplify genes, but we'll talk about how that works when we get a little bit more into gene cloning. Um, but first, I did want to talk about the limitations of PCR. And so, PCR is this really great tool, helped improve the efficacy of gene cloning and amplifying DNA, but it does have its limitations. It's not a perfect technique. And so, for example, one big limitation is that we can only amplify sequences of DNA if we know the sequences. And the reason for that is because those primers we design in a lab, and we design them based on knowledge of the sequence. If we don't know the sequence of nucleotides of the gene we're interested in, or that fragment of DNA, we can't design primers because primers have to be complementary to the sequence that we're going to amplify. And so that's a huge limitation. Unknown sequences can't use PCR with. Contamination is really easy. It is extremely easy to contaminate PCR. Any small little bit of tissue that falls off of you that you don't even see because it's microscopic can contaminate it. So, as like a hypothetical question, imagine we are actually working with patient samples and we are sequencing them, or trying to run them through PCR to see if we can detect if the patient sample has cancer-causing alleles. Um, if our own tissue sample falls into the patient sample and then we run it through PCR, it can either muddy up the results or even cause a false negative, and that your DNA gets amplified instead. And if that's the case, it's showing the results for you and not the actual patient. So if they have cancer, it can come out as a false positive. Really easy to contaminate. So a lot of care has to be taken. The accuracy. It is really accurate, but Taq polymerase does make mistakes. One mistake for every 20,000 new base pairs, it makes a mistake. So it is expected that every once in a while, running PCR, the sequences will actually pretty often when you think about it, but there will be mistakes in the nucleotide sequence. It's really easy to overcome that, but we won't get into the details of that. Just know that mistakes do happen with Taq polymerase. And then, of course, size limitation. So genes or sequences of DNA that can be amplified with PCR, usually there's a limit of around 2,000 base pairs, 2,000 to 5,000. Anything past that, it's with normal PCR, there are issues, and it's usually not feasible. And so, if you remember from the beginning of lecture on Tuesday, that dystrophin gene is about 2.2 million base pairs, way too big to amplify. So PCR would never be used to amplify dystrophin gene because it's just way too large. So these have to be relatively small genes. And a lot of genes aren't that small. A lot of genes are much larger. So we do see a limitation in that. Okay. Now, when it comes to applications, there are quite a few, but obviously molecular biology, it is a huge part of this field. And so it's just some examples. Um, let's say we wanted to look at genetic variation. Like, do we see that between two different subpopulations of a species, there are differences in their genetic makeup? And so let's get more specific with this and pretend maybe I'm interested in squirrels that are found in highly populated areas versus squirrels found in not very highly populated areas. And I want to know, is there a difference in their genome? Do we see that maybe they have some allelic differences, allowing squirrels in populated areas different personality traits or different behaviors, etc.? I can run PCR on both populations and compare them to see if there are differences in their sequences of sequences I know that are of interest. Obviously, diagnostics. I talked about diagnostics, how we can diagnose viral infections, how we can determine if a patient has a genetic disease like cancer or some sort of inherited disease. Uh, maybe we're interested in rare species within an environment. And so we want to know if a species is present in this specific environment, but this it's really hard to observe the species because it's so small and it's rare. We can take a sample of the environment to see if any of its DNA is freely floating somewhere. So if we can make primers for genes specific to that species, as long as we know the sequence of some of the genes within that species. Um, and then, of course, forensic science, which I'll talk about later, get to it a little bit later. PCR has been made much stronger with the advent of two techniques. One is um, real-time PCR or qPCR, and then the other one is reverse transcription PCR. And so qPCR, I think I talked about that a little bit before. But basically, what we're doing here is we're actually able to, in real time, while the experiment's going forward, quantify the amount of DNA present in our sample as the DNA is being amplified. And so this is a really useful tool to see, basically, give us a better idea of the amount of original sample and the amount of time we actually need to go through PCR before we have amplified it to its like limit. Um, and so I showed this image before. It is a graph showing us the relative abundance of DNA sample over time. And time in this case would be the number of cycles. And so each color here corresponds to a unique gene. So we can target many genes in PCR by using multiple primers. And so um, what we're seeing here is at what point was this gene amplified, and it gives us an idea of the original number of copies in that sample. The other thing is using reverse transcription. So normal PCR, when it was first developed, able to amplify DNA, and that's it. But some really smart researchers came up with some ideas, and they asked, hey, what if we include this enzyme called reverse transcriptase that we isolated from a virus? What reverse transcriptase does is it converts mRNA or RNA into DNA. And so what we can do is we can take a sample of an organism, isolate the DNA, or sorry, isolate the RNA, the mRNA, add the reverse transcriptase, and create cDNA, complementary DNA to those mRNA sequences. Then we run those cDNA through PCR. And so this was a huge addition to the toolbox of PCR because if we can get the quantity of mRNA transcript that tells us about the actual activity of the cell, what genes are active in that cell, and even at specific time points. So, for example, maybe I want to look at what genes are active in neurons in early development of an individual when they're in the womb or as a fetus, and compare that to what genes are active as an adult. that can tell us what genes are involved in cell differentiation for neurons. Um, because in early development, those neurons are still undergoing that differentiation, so they need genes associated with that. Whereas in adults, we'll only see genes that are associated with those adult neurons that are not undergoing differentiation. Um, yeah. And then, of course, the example of uh, RNA viruses. We can make complementary DNA of them, run it through PCR, see if a patient has a viral infection. And this is an actual diagnostic technique. Okay. Together, though, we can have real-time qPCR. Um, and so that's the case where our sample is of RNA, and we are quantifying it as we run through PCR. And it becomes much more useful in this case because um, typically when it comes to DNA copy number, that's somewhat stable since every cell contains two copies of every chromosome, meaning they have two copies of every gene with some deviations. But when it comes to mRNA, the amount of mRNA present in a cell is highly variable. It depends on the cell type, it depends on the time that it's been sampled, it depends on a lot of things. And so that tells us a lot more about the expression makeup of that cell, what's going on with that cell. Okay. And so, as an example question, um, what if we have a patient that has a disease? We can go with cancer as the example again. Um, and this patient's undergoing treatment, maybe chemotherapy, and we want to see the expression of tissue that's being affected by the cancer to see if we see a reduction in the expression of any alleles that are associated with cancer development. As treatment moves forward, if we see a decline in the expression of those genes that are associated with cancer, we can say that the treatment has been effective in remission um, uh, as far as it's being followed. Or if the patient has basically fallen back into the disease and the disease is starting to take over again, the cancer is starting to grow again. Uh, and so it can be useful for prognosis and monitoring treatment. Okay, quick concept check. So let's say you isolated a human sample DNA to see if they have a particular viral infection, and their cells. Let's say that it just happens that 10 cells were carrying one copy of the viral RNA. If you run PCR for 10 cycles, how many copies of the viral RNA would you have at the end? Uh, and so I just realized for some reason I accidentally put 30 cycles in the answer. But the way that this would be solved is you would multiply the number of original copies you have. In this case, it's 10, because one copy in the 10 in each of the 10 cells. 10 times 2 to the power of however many cycles you have. So in this case, I put 10 cycles. So it would be 10 times 2 to the 10th power. And that will give you the number. And let me calculate it right now real quick just so I can mention it. 10 times 2 to the 10th. And that gives us 10,000 copies in 10 cycles. And that's already a sufficient amount for it to be detected. Um, but again, PCR typically runs for a lot more cycles. I put 20 to 35 here, but I've run it even past that to like 40 cycles. But if we pretend we ran this for 30 cycles, instead the number would come out to be 10 billion copies. And so at that point, if we're not able to detect DNA after 30 cycles, it means that that sample, the DNA is not present, or the RNA, whatever it is we started with. And so if a patient, we ran their sample through PCR and we find that at the end it's not detected after 30 cycles, most likely they just don't have the infection. It's not present. Of course, they'll always say, um, that this doesn't rule out the disease, but it shows pretty high confidence that it's not present there. I believe the error rate for PCR is like, I don't know, it's less than 0.01% extreme confidence. And so usually it's not the only thing used for diagnosis, but it's a very strong tool for it. Okay. Okay. Second part question. What would happen if you ran PCR without a magnesium cofactor? Or what would happen if you ran PCR without the DNTPs? Um, and so you should probably pause it right now before you give the answer so you can try to figure out the answer yourself. Try to think about it a little bit. We want to start playing with the ideas here. Uh, Now, hopefully you paused it, and then you're coming back to see if you got the answer correct. Basically, what would happen is PCR just would not work. We would not get amplification of our genetic material. And that's because DNA polymerase, Taq polymerase, will not work without a magnesium cofactor, will not make copies of the genetic material. Uh, and then if we don't have DNTPs, we don't have the building blocks. It's like trying to build a building without, like cement blocks or without bricks. We need those building blocks for it to actually work. Okay. And so even though PCR is this amazing, great tool, it's so often used, like almost every genetic lab uses it. Um, it it doesn't mean that gene cloning no longer has application, and that it's just no longer used. Gene cloning does have application to this day, um, and that's because it offers something that PCR does not. And that's that with gene cloning, we can not only clone the gene, but if there's a protein product, we can also clone that. Um, and so PCR cannot possibly clone any protein product. It can do DNA, and then convert RNA to DNA and then copy the DNA, but it cannot do proteins. And so, as a really good example, insulin. We need insulin. It is one of the most sought or bought drugs on the market, um, or I should say, pharmaceutical products. Um, and the way that insulin is produced is by cloning bacteria with the insulin gene and then allowing them to produce protein product. And then we isolate the protein from the bacterial culture. Um, what we're looking at in this image here is these are really large vats in which they're brewing insulin. They have bacterial cultures in here. The bacterial cultures are producing tons and tons of insulin at once. And then the insulin is isolated from these bacterial cultures. It's almost like I don't know what to call them, but those brew vats that basically are brewing a bunch of beer inside, they're using the same concept that those vats that produce beer at breweries, they're growing a bunch of yeast culture, and the yeast is producing the alcohol. But in this case, we're growing a bunch of bacterial culture, and the bacteria are producing the insulin product. Um, and so I don't know why I circled around it so many times. But anyway, um, we're able to produce tons of insulin by this method. And this was a huge development on its own because for the longest time we were harvesting pigs. Harvesting pigs for insulin production. Obviously, that's way more costly. That's ethically concerning. These pigs literally grown for the whole sole purpose of producing our product and then just killing off, which is it's a whole horde of issues. Since we still technically do that to today with food and stuff, but I'm not going to get into that. Um, the point is, through this genetic tool, this development, this gene cloning tool we have, we've been able to make huge changes in not just for humans, but for other organisms, pigs in this example. Okay, so we'll go back to gene cloning so we can talk a little bit more about some specific details of how cloning works. How do we get a gene into a plasmid vector, which is this circular DNA here? And how do we get that plasmid vector into bacteria so that we can grow the bacteria with the gene of interest? And so the first part is we'll talk about this cloning vector thing. Usually a plasmid is taken, a known plasmid will serve as a cloning vector. And what we'll do is we'll insert the gene of interest into that plasmid or cloning vector, and then insert that vector into bacterium. And so every plasmid vector or cloning vector will have three important characteristics. They have to have these things or else it won't work. And the first thing that they need is an origin of replication. And so we see it in this image here, this origin of replication. If we do not have an origin of replication, oh, let me fix my seating. There we go. If we don't have an origin of replication, we're not going to get replication of the plasmid because that's what's identified by replication machinery to start DNA replication. So necessary. We need reporter genes. In this case, it's a selectable marker here. I'll talk about that a little bit more later, but this basically is telling us, did the vector take up the gene and did the vector get properly inserted into the bacterium? Which is why we call it a reporter. It's reporting the success of the experiment, the transfection. And then lastly, restriction sites. And we need these because this is where we're going to cut open the plasmid to insert the gene. And so again, plasmids are source of vectors. We'll isolate them from bacteria. So many plasmids have already been sequenced, and you can just purchase them now at this point. If you're trying to run one of these experiments, you just purchase them from a biotech company. But one of the real common ones is this PUC19 isolated, I believe, from E. coli. Um, but basically, what we're looking at here at the right is what's included in this vector or this plasmid. It has an original replication, an AMPR gene, meaning this gene provides resistance to ampicillin, an antibiotic. And then we have these restriction sites where we can cut open and insert a gene. Um, oh, and real quick, just there are so many different restriction sites that can be included within these uh, vectors or these plasmids. Okay, so how do we get the gene into the plasmid? Really simple. I think I mentioned this before a little bit briefly, but we treat our plasmid as well as our foreign DNA separately. We treat them with restriction enzyme digest, meaning we add restriction enzymes and they'll cut at those restriction sites. Cut here, 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 for our plasmid as well as our foreign DNA. This will cut out the gene of interest, and for the plasmid, it just basically cuts it open. Um, when we mix them together into the same solution, what we see is that the gene of interest will be inserted into the plasmid via complementary base pairing. Those sticky ends are complementary to each other because the same restriction site is being used. And so it'll insert, attach, then we can add our DNA ligase, and it'll seal up that plasmid with the new gene. Okay. Oops. Yeah, I forgot I had that written here. And so we did that restriction enzyme digest. The annealing part is where we actually allow the gene to enter into the plasmid. And then we have our ligation, where our DNA ligase will come in and seal it up. Now, once we make the plasmid, oh my God, sorry. I hear like neighbors and stuff. Anyway, once we make the plasmid, we want to insert that plasmid into bacteria, and then we want to grow those bacteria. Maybe you just want to grow the gene, or maybe we also want to grow the gene and its protein product, like with insulin. And so, how do we get it into the bacterium? We take a bacterial culture, we mix our sample of our transformed plasmid, and we allow the bacteria to take up the plasmid. Now, some bacteria naturally will take it up, and that's all you have to do. And then you just plate them, grow them on a plate, and then take colonies that have properly taken in the plasmid, and then grow them, isolate them, and grow them separately to produce your product. But a lot of bacteria don't take them up that easily. They need help, and so we treat them with heat shock. Um, yeah, I should mention it here. We permeabilize some bacteria with this heat shock treatment. And what that does is it makes it easier for the bacteria to take up the plasmid. Once they take it up, then we go to the next step where we take our sample and we plate it, and then we allow it to grow. We uh, identify any colonies that have properly taken up the plasmid, take that sample to isolate it, and grow it in a new broth. to make our product. So, um, how do we know that a bacterium has properly taken in the plasmid? So we have this step here. I'll go back real quick. We have this step where we isolate the colonies that did take in the plasmid. How do we know that our colony properly took up the plasmid? How do we identify which colony did? And that's where those reporter genes come into play. And so with our reporter genes, they technically come in two different flavors. And so one of them is the gene confers antibiotic resistance. The other type of reporter gene is that it provides a fluorescence or some sort of pigment. It produces a pigment that causes color, so that we can visually see it on our plate. Um, and so um, let's go ahead and look at this image a little bit closer. And I'll talk about that in a second. Um, Let me go ahead and move my face real quick. What we're looking at here in this image is we have our plasmid vector with our AMP-R gene, that's one of our reporter genes, and then we have what's called our LAC-Z gene. This is also a reporter gene here. Our restriction sites are inside this one of these reporter genes. And so our gene of interest, our foreign DNA, is going to be inserted into that reporter gene there. And don't worry about what the LAC-Z gene does right now. Just know that when this foreign DNA is inserted into the reporter gene, this gene is no longer functional. The LAC-Z gene is basically turned off. But we still have this other gene here that provides resistance to ampicillin. And so we'll start with this one because this one's a lot easier to understand. When we mix our plasmid with our bacteria, we can take it and then we'll take those bacterial samples, grow them on a plate, and that plate will have ampicillin, the antibiotic. If our bacteria did not take up the plasmid vector, they will not have the ampicillin resistance gene, and they will not grow on this plate. If bacteria did take up the plasmid, they will have the resistance gene, and they will grow here. Sorry. Um, no, that's only telling us one thing. That's telling us that the bacteria growing here, they did take up a vector, but we don't know if the vector that they took up actually has the gene of interest or if it's a vector that wasn't properly transformed, because that does happen. Not every single plasmid is going to take up a gene. So we need some way to know if bacteria took up a vector and if that vector that it took up does actually have the gene of interest. And so that's where this LAC-Z gene is going to come into play. What LAC-Z does is it breaks lactose. Lactose is two pieces. It'll break lactose into glucose and galactose. But instead of adding lactose, what researchers can do is they can add a modified galactose, where it's a galactose with this chromophore attached to it. And it uses the same chemical bonds that we see between galactose and glucose. And so what we will see is if a bacterium took up this plasmid, this gene doesn't work, so this X-gal, that modified galactose, will not be broken apart. It will be left alone. If that's the case, no color shows, and we get white colonies. If a bacterium took up the original plasmid that was not properly transformed, it will have a functioning LAC-Z gene. That LAC-Z gene will break down this X-gal, it'll break it apart, and when X-gal is broken, that added chromophore will actually emit a light. It will emit a blue color. And so bacteria that took up this unchanged plasmid will see them as blue colonies on our plate. And so these are colonies we don't want. Colonies we do want in this case are those white colonies that weren't able to break down X-gal because their LAC-Z gene wasn't actually working. And so that ensures we're taking the right bacteria. Okay, now let's go back a little bit to gene cloning, the part where we're talking about getting a protein product. A lot of times, well, I should say sometimes We can use bacteria to get a protein product because the gene of interest is pretty small, and insulin is a really small gene, so it's not that big of an issue. Um, and on top of that, sometimes those genes require very little processing or mRNA processing, meaning um, we don't need any introns cut out, or we don't need any special treatment to the mRNA before it's turned into a protein. But for most eukaryotic genes, we need mRNA processing to properly cut out introns to get our properly functioning protein. And so the issue with that is that bacteria can't do that. Only eukaryotes can do that. So how do we clone a gene in a eukaryote so that we get proper mRNA processing? Um, so let's move on to the next bullet point. What we can do is we can use, well, one retroviral vectors we can use, but the other one I wanted to talk more about was genes or chromosomes that we have in eukaryotic organisms. And so what we can do is we can take a vector from a virus or bacterium, and we can add um, things that that vector needs to properly function in a eukaryotic cell, like a yeast cell. And so what I mean by that is a chromosome in a eukaryotic organism needs a couple of things that bacteria won't have. Those telomeres, which we see in gray here, we need those telomeres. And then on top of that, we need these, where is it? Our centromere. We need our centromere here. Um, those are needed for the plasmid to function within the yeast. Once we have all those pieces necessary in the plasmid, we can put it into a yeast, and a yeast will be able to take the gene and transcribe it and then translate it and produce the protein product. It will be able to undergo the mRNA processing so that we get a proper protein product where introns were cut out, and it was spliced together properly. And so this yeast artificial chromosome, really commonly used, we're seeing it here. Um, and that one has been commonly used to help grow protein that we can't properly grow in bacterium alone. Okay. Okay. And so applications of gene cloning, we saw insulin was a great application of that, but some even more intricate, uh, intense ways that gene cloning have been used is in the case with this soil bacterium, Rhizobium radiobacter in agriculture. And so this bacterium, Rhizobium radiobacter, what it does is it causes what are called crown galls, and we're seeing them here in this image. These crown galls, they're like tumors. We get this unregulated growth here. But what it does is it helps the bacterium. And so these bacteria, they contain this plasmid called the TI plasmid, a really big plasmid. Um, and when the bacterium infects the plant, the TI plasmid is transferred from the bacterium to the plant upon this infection. And what the plant does is it incorporates this TI plasmid into its own genome. And then the plant starts producing the protein from the TI plasmid. And what these proteins do is a lot of them are enzymes that actually help the radiobacter grow, the bacterium. And so it supports its own infection this way. Researchers have taken this TI plasmid and they have transformed it by adding a toxin, this Bt toxin, which is an insecticide. Insecticide meaning it kills insects that damage crops. And so they've inserted this toxin, and on top of that, they've removed or knocked down any of those genes that are associated with crown gall disease. So it does two things. It prevents the disease of the plant. And on top of that, these plants are going to start producing a natural insecticide, the Bt toxin, that is harmless to humans. And this is used in a lot of different plants, including tobacco, cotton, tomato, corn, etc. It's really common. Guaranteed, you've eaten plants that have this TI vector or plasmid. Uh, oh yeah, so some crops here. What we're seeing in this image is we have some crops here that do have the insecticide, that TI plasmid, and some crops that do not have it. And so we can see there's a complete difference in the yield, the crop yield between these two sections here. Okay, quick concept check. I would pause it now, try to answer the question, and then come back to see what the answer is. So, hopefully you paused it. The question is, how is a gene inserted into a plasmid cloning vector? Um, Again, there's a few steps there, just really three. One, you do restriction enzyme digest of our foreign DNA in our plasmid vector. We'll cut out that gene and then open up the vector. Then we'll mix them together and allow annealing where the gene inserts into the plasmid vector. And the way that that happens is those overhangs of the plasmid will form complementary base pairing with the overhangs found in our gene of interest. And that's because the same restriction site was used. They'll anneal. Then we'll go in the third step of ligation where we add DNA ligase and it'll seal up those nicks. Hopefully you got something like that. Okay, so all these techniques I've talked about so far on Tuesday and so far today, they require that we know the gene of interest. We know the sequence of it, and we know a lot of information about it. Um, but what if we don't actually know the sequence of our gene, or we want to identify genes? Like maybe we're curious what genes cause, if there are any, um, cognitive impairment found in something like ADHD or autism. Um, what are the gene sequences? How can we identify them so that we can learn more about these genes and maybe even in the future potentially fix these genes using gene therapy? Um, there are a few different methods. The oldest probably is by using what we call or creating what we call a DNA library. And so this I'll go over briefly. It is it's still a thing because it's a precursor in some other like techniques, but otherwise as a standalone I would say it's much less common or gone out of vogue. But basically what a DNA library is it's a collection of DNA fragments that encompass the entire genome of an organism. So what I mean by that is I guess we should just bring up the image, is that when we wanted to sequence genomes or entire chromosomes, large pieces, historically, one of the first techniques to be used was this shotgun sequencing, where we introduce this large sample of DNA, like a genome, to restriction enzyme digest. And these restriction enzymes, they're going to come through and make random cuts throughout the genome. And what we end up with is a bunch of fragments of the genome of different sizes. And so these fragments, what can be done with them is we can make these high dilutions. And what I mean by high dilutions is we really dilute our sample of DNA far down, and we introduce that sample of those fragments, that diluted sample, to a bunch of vectors. And what we're doing here is we're diluting our sample so that when we introduce vectors into the sample, each vector should take up only one fragment. There's still going to be vectors that take up more than one fragment, but the vast majority of vectors, since there's so many of them, they will only take up one fragment into the vector. And then we can take these vectors, we can isolate them through dilution again, isolate these vectors into their own individual sample tubes. And then when we do that, we can take samples of each of these tubes and expose them to individual colonies, meaning that this sample here, this tube, contains this one fragment here, and we're introducing to this one colony here. So now when we grow these bacteria, all of them are going to grow this vector here with this specific fragment. What this does is we have a bunch of plates, and each plate contains a unique piece of the entire sequence or genome of interest. Then we isolate some samples of the bacteria. Let's say we can take this plate, we'll isolate their plasmid again, and then we will sequence the plasmid to see what the sequence of the fragment is, since we know the sequence of the rest of the plasmid here. And so we'll get the sequence of this fragment. And then what you end up with is a bunch of pieces. We have these sequenced fragments, and then we map them to each other. And so this is a little bit simplified. So you might be wondering, how do we know this one belongs here and this one doesn't belong way over here or something? And the reason for that is because if you run a bunch of samples of that, whatever it is, let's say it's like human skin cells, um, when you run the restriction digest, we will get random cuts in different areas for different samples. So maybe we'll get this fragment here, but maybe in another sample, this fragment is actually two pieces instead, two small pieces, and these two are connected. And so what that means is what you have is fragments that if we're looking down here, some fragments that overlap different parts here. So maybe we'll have a fragment that overlaps a piece of this first fragment and this second fragment. And if they're overlapping, we'll see the sequence matches to these two here. And basically, what it's like is you're taking a bunch of puzzle pieces and putting them together so you can get a clearer image. All those overlaps will allow us to see what the contiguous sequence of DNA is. And hopefully, or maybe to better help understand that, um, if we have a fragment that overlaps these two pieces here in the middle, so hopefully you're kind of following my mouse right now. Um, if we have a fragment that overlaps here of this long piece and then here as well, this single fragment, we know that the sequence is complementary to this piece and to this piece, and that it is together, they're continuous. That means that only this piece can come after this piece. No other piece can, because no other piece will be complementary in sequence to the fragment that overlaps these two. And so you'll have a bunch of fragments overlapping, allowing you to make a map of this entire genome. Obviously, this is requiring a bunch of computer coding and a bunch of computational analysis that I'm not going to go into because that is a much more advanced class. Okay, so um, we can do cDNA library, which in this case, instead, unlike just a normal DNA library, cDNA, again, that means complementary DNA. What we're doing here is we're actually taking mRNA, running it through reverse transcription, um, and then taking those pieces, those RNA, those cDNA sequences, and running them through the same process of the gene cloning. And now what we'll have are let me go back to this image real quick. Here in these plates that we would have from this cDNA is each plate represents a unique mRNA transcript, which in another way we can think about that is that each plate represents a unique protein or a unique gene. And so this is useful in that when we're creating a DNA library, it's the whole genome, including non-interesting stuff like non-coding DNA that maybe has zero function. In this case, when we're making a cDNA library, we have the advantages of we're looking at interesting stuff. We know that these are genes because they were converted into mRNA. And we know that there must be a protein associated with these mRNA transcripts. Does not contain introns. Another good thing that's usually introns are not super interesting. Caveat to that. I'll mention in a second. Um, and then again, it's enriched for actively transcribed genes. So what that means is if we take a sample of skin and we run it through this process, any genes that are silenced, meaning that they're not active in the skin cell, we're not going to see them in our cDNA library. And so all the genes found in that sample are associated with that cell. In our skin cell, we'll see a bunch of genes that are specific to skin cells, only found in the skin cells, and then a bunch of genes that are needed for normal cell function for like every cell. If we take a sample of skin cells, and then we take a sample of like neurons, for example, and then we can compare the content of cDNA of both of them, we can see if there's overlap. Are there genes or sequences present in both samples? If there are, we know that all of those genes are probably necessary, for the most part, normal cellular function. If there are genes only found in the skin cells, those genes are needed for skin-specific functions. Any genes found in the neurons, those are all genes needed for neuron-specific functions. And so we can see how this can be extremely useful when we're looking at different tissue types or even at different stages of development. Okay. Disadvantages, this is where that caveat comes in of not containing introns. Um, Sometimes introns do have interesting stuff like splice site information. Sometimes, other DNA that's not part of the actual gene itself, the coding part at least, um, won't be included, like promoters. We won't get the promoter of a gene, and that's because our mRNA transcript does not contain the promoter sequence. Functional RNA, like our tRNAs or our piRNAs or microRNAs, etc., that won't be included. And that's because this is enriched for mRNA. And then, of course, regulatory elements. Those regulatory elements like distal promoters or silencers or enhancers, things like that won't be included. And then again, this is also a caveat that this depends on the tissue or cell type. Um, if we're asking a question about, let's say, neurons, for example, we want to isolate neurons only. We can't unintentionally include other cell types because that can really confuse our findings. Okay. General process here. This image is horrible. I'm so sorry, but it's really hard to find a good flow through image for it. I think I just need to create one myself. Um, but what we have here is we have isolation of RNA, collection of the mRNA, putting into its test tube, submitting it to reverse transcription so that we can create our cDNA, insert the cDNA into plasmids, take those plasmids, insert them into bacteria, transform them, grow our bacteria, and then take our bacteria or our plasmid out and sequence it. Isolate the plasmid, the DNA, and then sequence that so we can get actual sequences of those mRNA transcripts. Okay, quick concept check. Again, this is one of those points where you should probably pause it real quick and then try to answer it, then play it and see what I tell you the answer is, to see how close you were, to see if you need to work on your understanding or whatever. How can researchers isolate mRNA from other types of RNA? So in this, cDNA library making, for example, or in our uh, yeah, we'll just stick with that. If we're trying to make a cDNA library of our mRNA, how are we supposed to isolate mRNA from other types of RNA like tRNA and rRNA? Is there anything that's different between mRNA and these other types here? Anything that's unique to mRNA? That question, what might distinguish mRNA from these others that can be used to isolate mRNA from them? And so you need to try to think about what is different, what is unique to mRNA. Let me go back to transcription real quick. So we transcribe our mRNA. What happens between transcription and then translation? And then I think that's the big part, right? That only mRNA is translated. So what is different? What happens to mRNA between transcription and translation that might make it unique? And if that's still not helping, then just go ahead and continue watching. Um, the way that we can isolate mRNA from these others is that mRNA has that poly A tail. There are two unique things in mRNA. There's that 5' cap, that methylguanine cap, and then we also have that 3' poly A tail. That is not going to be present in these types of RNA. So we can insert probes that are complementary to the poly A tail, or that specifically grab onto that methylguanine cap, and it'll pull the mRNA out of the rest of these other types of RNA. We can wash away these other RNAs with some salts or some other types of solution. And then when we wash it away, all we are left with is our mRNA transcripts. So if you got that, awesome job. Okay, so once we have created a DNA library, we have this entire sequence of DNA that includes some coding regions or genes and some non-coding regions like introns and then anything outside of genes. Um, how can we screen our sample set for sequences of interest? So What if we're wondering, does this bacterium produce this endospore gene? Or sorry, this endospore protein that's found in some other somewhat related bacterium. And this endospore is clinically significant, meaning that if it's found in food, it causes severe food poisoning, like E. coli or something. Um, we want to know if our sample species or tissue, whatever it is, contains the gene of interest. What we can do is we can run this nitrocellulose assay here, where we have our colonies of growth. We add this nitrocellulose filter on top of our colonies, and what happens here is genetic material will transfer from the colonies into the nitrocellulose filter. We'll remove that filter, and it forms like a map of our colonies. We'll put that filter into a tube in which we add probe, and that probe is complementary to that sequence we're interested in. We'll add the probe into our sample, and it'll bind or hybridize with sequences, um, the sequence of interest if it's found in any of the colonies. Now we have a filter paper where we see highlighted any colonies with sequence of interest. And then we can map that filter paper back over our original plate and we can isolate those colonies. And so we can see them here. These are the ones that contain the sequence of interest. So let's go ahead and isolate them. Okay. And so again, probes are kind of like primers in a way in that they bind to sequences of interest through complementary base pairing. And so we need to know the sequence of interest if we're going to run a probe. And so how do we know the sequence of interest? How do we know the actual sequence of DNA? So if we're in the previous example where we're wondering, does this species of bacterium contain the gene for an endospore that causes severe food poisoning? And this gene we know is in some other somewhat related bacteria. How do we know the sequence to create a probe for the gene or the species we're interested in, or the strain? What if we don't know the actual sequence in that species? And so there are a couple methods that can be used. One is that, like I mentioned, if we know another species or strain contains the gene or sequence of interest, oftentimes we can just use that sequence as a reference to create a probe. And that's because probes don't need to have 100% binding not accuracy or sequence accuracy. There can be a couple nucleotides here and there off, and we'll still get hybridization. And so we can use the gene from that similar or related species. Um, if the protein sequence is known, so maybe we're interested in a protein, and that protein has been isolated from the bacterium and its sequence is known, we can use that protein sequence to predict what the DNA sequence of that protein was, and then create a probe based on that predicted sequence. Um, and so this might sound a little bit counterintuitive because you might be wondering, well, why would we know the protein sequence if we don't even know the DNA sequence, the mRNA sequence? And the reason for that is because, one, we learned how to sequence proteins before we learned how to sequence DNA or RNA. And so we have a bunch of libraries of protein sequences already known, but maybe not the actual gene sequence. Um, and then two, because of that, it is sometimes easier to just sequence the protein first and then run backwards with that, creating a probe from it. That said, creating a synthetic probe based on the protein sequence is still a little bit hard. And that's because if we know the protein sequence, we technically don't know the exact nucleotide sequence. And that's because the codon redundancy. We know that, for example, um, proline has four different possible codons that code for proline. So how do we know which one is the correct codon that's found in the original DNA sequence? And so because of that, usually what happens is multiple different probes are made trying to account for all the possible sequences for that protein. And then all those probes are added. And then one of them is going to work. And then the third method is um, getting a protein to make, sorry, getting a bacterium to make the protein, sequencing the protein, and then making the probe based off that protein. which sounds like a whole workaround, but sometimes it's just more feasible. This image at the right, all we're looking at here is um, an illegal nucleotide synthesizer. So this is basically an instrument or a machine, and what it does is we supply it with reagents. Usually the reagents contain a few things. One, enzyme like DNA polymerase and its cofactor magnesium, DNTPs, um, and then some other, like primer sequences. Actually, not primers in this case, but we have some other reagents that are necessary for um, us to be able to actually create the primer itself or the probe itself. So we're having enzymes in this instrument take the building blocks to make the probe that we want or the primer that we want. And they're super duper easy to use. You have this little GUI interface here, this little computer where you can input the settings, the sequence of your primer probe that you want, and specifics about it. Okay. On top of all of these techniques, we also have some other really powerful techniques that tell us more information. So, for example, maybe sometimes we're curious about the presence of genetic material inside the cell itself in the moment, or inside of a tissue, or inside of a whole organism. And so we can run what's called in situ hybridization, or in situ, hybridization. And so what in situ refers to is this is a Latin term for meaning inside of the cell, in the sample. And so what we can do is we can put probes into a sample, into a tissue or a cell or an entire organism, and allow those probes to bind with the sequence it is complementary to. And so we call this in situ hybridization because we're allowing probes to hybridize with the sequence of interest inside of the cell. And so oftentimes fluorescent probes are used, and we call this fluorescent in situ hybridization or FISH. You've probably seen that term before, but you're like, didn't really pay attention, just glossed over it. But it's a really commonly used technique. And so this is an image here of what that looks like. And I'll move my image over again. And so what we're seeing here is we have chromosomes isolated from a cell, and just in case you aren't already familiar with this, these chromosomes were isolated during metaphase of mitosis, when the chromosomes actually take their full-on shape. If we add fluorescent probes to our sample here, and then we denature these chromosomes, meaning we allow the two strands of DNA to separate, the chromosomes will maintain their structure, but within the chromosomes, the strands separated, this is going to allow our probe to insert in complementary base pair with the sequence of interest. And so in this image, what we're looking at is we have a probe. In this case, it should be for one marker on chromosome 7. Um, and we have a probe for one marker or one gene. In chromosome 7 here, what we're seeing is a duplication. And the reason we know that is because in this chromosome, we see the probe highlighting two spots near the centromere and then at the ends near the telomeres. This gene should only be in one spot on the chromosome. And so this is a duplication of that gene. On the other hand, here, what we're seeing is instead we have a deletion in one of our chromatids. We only see one fluorescence mark in this chromosome here. So we see a deletion here instead. This chromosome or a chromatid copy does not have the gene. There is no fluorescence there. Okay. And we can even tag material inside of whole tissues or organisms. And we can tag mRNA, which again tells us more information about the activity of genes in different tissue types or different cell types, or in organisms at different developmental stages. And so in this image here, in part B, what we're seeing is this is a fly embryo. And in this case, what they've done is they've hybridized or targeted specific mRNA sequences associated with development of specific anatomical features or organs, like the head region and then the tail region. Okay. And so for a lot of genes that have important functions, we don't have a lot of information on them. So, with genetic diseases, some of them we still don't know what gene is actually causing it, or maybe there's a bunch of genes causing it, and we don't really know which ones um, contribute most, or have, or if there's any interaction there. So, how do we identify and isolate any genes of diseases that are still not well understood? Um, and there's a few different mechanisms that we can do, but one of them is what's called positional cloning. And so, what we're doing in positional cloning is we are isolating genes based on their position in the genome, what chromosome they're located on, and where. And the way that we can do this is by using genetic markers. And so, for the human genome, we have a ton of genetic markers interspersed randomly throughout the genome. And so if we're looking at specific chromosomes like chromosome 1 versus chromosome 7, there will be unique markers that we have spread out through the chromosome. And so, what we can do is we can look at the inheritance pattern of the disease-causing allele or gene. So we have a pedigree here, so we can see how a gene or disease is being inherited. And then we can also see how specific genetic markers are inherited across this pedigree. And so if we see that a specific genetic marker is following the inheritance pattern of the gene of interest, um, then we know that the gene of interest is close to that genetic marker. And so in this little image here, we have hemophilia, for example, the inheritance of hemophilia. Um, we have our carriers, and then there are individuals that are actually affected that are female, and then are affected males. We can see that this is a sex-linked trait because females are very often carriers but not affected. So this is probably an X-linked recessive trait. And so males are primarily affected. That already tells us a little bit of information, makes it easy. I wish I used a harder pedigree. Um, but in this case, we already know that the gene is probably on the X chromosome. But what we can also do in this case is we can use markers on the X chromosome and see if their inheritance pattern is matching that of this disease. And that's because this disease, its inheritance pattern isn't going to be perfect. Um, and if there's any marker on the X chromosome that's matching the inheritance pattern here perfectly within the family, then we know that that marker is very close to the gene of interest. And so that already gives us like, now it's no longer a needle in a haystack. Now it's like we've removed 90% of the haystack and we have like a small pile of hay and the needles in there. And it's going to make it like 10 times easier to actually find the gene, but we haven't actually located it just yet. And so we need to add some extra techniques to that. This is everything I already mentioned, so let me go ahead and move forward. On top of that positional cloning, we can use this technique called chromosome walking. And so what this entails is, again, we're using gene cloning in this case, where we take the sample, the human gene sample, that small area, the little pile of hay that we've isolated it to, take that, insert it in, and submit it to restriction enzyme digest, and then insert it into bacteria to create colonies to clone it, all the different fragments. Um, and similar to our DNA library, what we will hope is that, let me see, make sure I'm not missing anything here. Yeah, okay, so I'll explain it here. What we're hoping for is um, we'll make a little DNA library and we'll also have overlapping pieces, just like I had mentioned previously. And with these overlapping pieces, what we can do is here's our marker. This blue one serves as the actual marker, the known sequence that we've included, that we know the location on the chromosome. We'll take the marker, the fragments of the marker, and what we'll do is we'll sequence it real quick and then create a probe that is complementary to a portion of this marker, an overhang part. So here, the blue part represents the actual marker, but we'll have some fragments that include the marker and a little bit extra um, a portion of the DNA. And so this fragment includes the marker and some more. And hopefully, and what happens is this extra piece that's not the actual marker, this will overlap with other fragments. And so when we sequence this and make a probe, and hybridize it with our clone A and prove that it does hybridize. Now we can take that probe and submit it to all of our other colonies and all of our other plates. And if we see any hybridization with any plate that we have, then we know that it contains a fragment that overhangs with this segment here. And so now we have a plate where we see the probe shows fluorescence or shows hybridization. We can say, okay, this plate contains our overlapping sequence here, clone B. We'll call this clone B now. Now, with fragments of clone B, we'll make another probe complementary to this, the end of this clone or a fragment, and this should overlap with other fragments found in other colonies or plates. And so we sequence this, make a probe, prove that it binds. Now we take this probe and submit it to all the rest of the plates. Great, we found another plate where we're getting hybridization. This one contains an overhang that overlaps with clone B. We'll call this clone C. Now let's sequence this, form a probe again for the end, take that probe, put it in all the rest of the Petri dishes or colonies. Um, we get hybridization again. Great. Now we know this Petri dish or this plate contains bacteria that have the fragment overlapping with our clone C. We'll call this clone D. And we'll keep doing this until eventually we have a fragment that does contain the sequence of interest. And the way that that can be identified now at this point is it a gene? When we sequenced all of this, we make a very long continuous sequence by getting the sequence of each of these and matching them up. When we look at this long continuous sequence, do we see any promoter regions, any consensus sequences of promoters? If we see them, for example, here, promoters show up here, we know there's a gene here. Is this the gene of interest? Um, And then after we've located that this is in fact a gene, we can go ahead and sequence it and then compare it to other closely related species that have sequenced genomes to see if it matches up with any of other genes to help identify the function. If we still don't know it based on that, there's some other techniques I'll go over in a little bit later. Okay, so we've identified a sequence we're interested in, or we want to know more about a sequence. How do we get the actual sequence of nucleotides of a gene or a region of DNA or mRNA? That's where DNA sequencing comes into play. And so DNA sequencing, in this case, we're reading the the genome, or we're reading the sequence of DNA, the fragment, whatever it is. And the way that it works is it relies on these modified DNTPs here. And so on the left, what we're looking at, this is a normal nucleotide or DNTP. It has its OH and its H. Remember that this OH is used to create or to help grow the DNA strand. A new nucleotide is going to come in here, and its phosphate group is going to bind to the O here and remove this H group and remove an oxygen there and form water. But if this OH is not there, like this H, we cannot add a new nucleotide on. So if this is incorporated into a growing DNA strand, growth will stop. No more growth. And so that's the key to how sequencing works. And so the first form of sequencing that was developed was called Sanger sequencing, and that was developed by Frederick Sanger in the 70s. And so this is still commonly used to today. And so this is a pretty big development. And what Sanger sequencing again relies on is these ddNTPs, these modified nucleotides. So let me just go to the next image and we'll see how this is working. Ignore all of this for now. Don't read any of this. Let's just go through the image and what's going on here. With sequencing, it uses PCR, but there's special changes. So what we have at the top of the image is we have our template. Basically, this is our sample of DNA that we've taken from whatever it is, our organism, our tissue, blah, blah, blah. We've added our primer, and we've made copies of our, whatever the gene of interest is, or the sequence of interest. We made a bunch of copies of it and inserted it into a new test tube. We still don't know the sequence. All we know is we made a bunch of copies of it. We take this sample, take aliquot portions out into four new test tubes. So now our copied gene or DNA has been added to each of these four test tubes here. But now in each of these four test tubes, what we've added is a unique sample of a DDNTP. So in this tube one, for example, we've added DDATP, a modified adenine. In this tube, we've added a modified cytosine, DDCTP. And here, a modified guanine, and then here in a modified thymine. Now we can run PCR on each of these samples and then run those samples through gel electrophoresis. Where those DDNTBs come into play is during PCR. During PCR, DNA polymerase is going to make copies of our template, but every once in a while, at random, it's going to add in one of those DDNTPs. And so in this first tube, it's adding a DDATP at random. And when that ATP, this DDATP gets added, the RNA polymerase releases and we get stopping or halting of the growth of that particular copy or that copy of the template. And so at the end of PCR, what we'll have, oops, let me go back. What we'll have is a bunch of different fragments of different sizes where each stops where that DDATP was added. So notice that we have a very short sequence here at that second adenine. We've added a DDATP, so growth stopped. And that's, complementary to the T here in our template. At the third thymine here in our template, we add our DDATP, and now growth stops here. Same thing here. At the end, we have another thymine, so when DNA polymerase comes in and adds that DDATP, we get stoppage at the very end. Same thing happens for every tube, but where it stops, it's different because of what DDNTP was added. Here, instead, in the second tube, we're stopping at every guanine. Every guanine we stop. And now we have fragments where at the end of the sequence we have a cytosine. Here we're stopping at every cytosine. So the first one, this second one, third, and fourth. And now what we have are a bunch of fragments that technically span the entirety of the template sequence. If we take each of these samples and run it through gel electrophoresis, so here is our lane that includes our sample that only had DDATP. It'll run across here. We'll take our sample here, put it into the lane for C, where we added our DDCTP, G, and then T. Because each of these differ in length by one single nucleotide, we have to run it through a special gel in a gel electrophoresis that separates nucleic acid size by a single nucleotide. And so our shortest sequence, this one here, not even showing up. Oh yeah, sorry. Our shortest sequence here, this G, is going to travel the furthest, and that represents the very start of the sequence. Our longest sequence, which is this one here that contains an A at the end, travels the shortest distance, stops here. Each fragment of each different size is going to travel down to a certain distance, but what we'll notice is that the lanes will tell us what position in the sequence that is. So this smallest fragment traveled all the way down here. That was our G lane. So we know that the first sequence is a GC pair, the first nucleotide. The second furthest distance is from our A here. So we know the second position is an AT pair. The third position we know is a TA pair because it ran down the T lane. And we just keep doing that to add on every single nucleotide in the sequence until we have the full sequence read out. And so this is a long process requiring multiple steps of PCR. I mean, it's not that long anymore because of multiplexing, but anyway, we'll move forward. This is just another summary of that, but what I want to highlight from this image, and I'll move my face over again, is that the main output of this Sanger sequencing is what's called a chromatogram, which we're seeing here. And what a chromatogram is doing is our sample of PCR is also run through a fluorescent light or a laser, and fluorescence is emitted because of the DDNTPs. And for each uh, nucleotide position in the sequence, we're going to get a fluorescence reading. And that fluorescence reading is going to be unique to each nucleotide. So what we're seeing here is yellow represents G, green represents A, blue represents C, and red represents T. Oops. Yeah, represents T. And so we have this little visual graph of our sequences. Okay, quick concept question. Pause it, try to answer, and then I'll tell you the answer. Did you pause it? Good. You better have. No, I'm just kidding. Okay, that was dumb. All right, so the answer in this case is I should have double checked what the answer is. Oh yeah, D. The absence of a 3' hydroxide group on the DNTP prevents addition of a new nucleotide. So that was key to sequencing, Sanger sequencing. Another concept check. This one I think is a little bit more of a brain teaser. Maybe some of you will be able to get it, maybe not. But what we're seeing in this image here is a sample of a patient and a sample of a mother, their mother. Um, and we have a chromatogram reading of both of them. But what's interesting is in the patient, at this looks like the seventh position, we have a single blue peak. And in the mom, instead, what we have are two smaller peaks, one red and one blue. And so the question I ask is, why do you think we have two small peaks here in the mom, only one big blue peak here in the patient? What could potentially be going on there? Um, And so just also to add on to that, here's a key showing which color represents which nucleotide. So blue is cytosine, green is adenine, thymine is red, and then guanine is black. And so go ahead and pause it, try to think about it, come back for your answer. Okay, so hopefully you paused it. And so the answer in this case is that for this particular gene, the mom is heterozygous. She has two different copies of the allele, and her two different copies are different in this base position or nucleotide position. And so one of her alleles contains our cytosine guanine pair, and then one of them contains a thymine adenine pair instead. And so she is heterozygous for this allele at this particular point. Whereas the son is homozygous and only has, in this case, it shows up as a cytosine, so a GC pair here. And so, if we're thinking about hemophilia, for example, that X-linked trait, uh, we can see that the mom was probably a carrier, and the son was affected, most likely. Okay. So, Sanger sequencing is really great, but it has its limitations as well in that it's really only applicable if we have very small samples. We're looking at very, specific genes or small genes, and we are only looking at just a handful, maybe a couple genes. But very often we want more than that. We want more. We want to know the entire sequence of the genome, or we want to know all the mRNA transcripts being produced in this cell or in this tissue. And at that point, Sanger sequencing becomes completely impractical. Um, and so that's when something else called next generation sequencing comes into play, where we're able to sequence large amounts in parallel, making it quicker and cheaper. And so um, there are different types. We have whole genome sequencing as a type of NGS, transcriptome sequencing or RNA-seq, another type of NGS, and then RAD-seq, which is using restriction sites, um, another type of NGS. And so with whole genome sequencing, we're getting the whole genome. That's including coding genes, non-coding material, mitochondrial genes, chloroplasts, etc. Our transcriptome or RNA-seq is giving us all of our RNA, which can be mRNA or any of our functional RNA like tRNA or rRNA, or any RNA that doesn't code for anything, and it's just like our piRNA, microRNA, etc. Proteome technically considers all the proteins present in a cell or sample. And so this and this wouldn't necessarily be considered part of next generation sequencing. Okay, and so there are different platforms, but by far the most popular for maybe even to this day, I haven't checked in a while because I haven't done research in a while, is Illumina. Illumina sequencing. They have their own platform, their unique form of sequencing. And so there's a video here, and I highly recommend you watch that now. So you might want to pause it, just go and watch it real quick, and then come back. Um, it should be in your copy of the PowerPoint. Um, but let's go ahead and move forward. The way that Illumina sequencing works is it's a lot similar to Sanger sequencing, where we usually have this, glass slide, and it has a bunch of small probes already attached to the slide. And those probes, they might contain primer, and when you add a DNA sample, your sample will hybridize with the probes or the primers. And so you can have a bunch of different probes or primers or a bunch of different sequences. Um, but usually before this, there's a whole nother step called barcoding, and I don't even want to go through that right now. But when your DNA random fragments bind to those probes or primers, we can then add nucleotides in using DNA polymerization in steps. So reaction is allowed to continue real quick, and then a nucleotide's added, and then reaction stops. And then after that moment, when the nucleotide's added, the computer is able to get a fluorescence reading of which nucleotide was added in that moment. So nucleotide's added, you freeze it to get fluorescence reading, then you allow it to go forward again. New nucleotide added, freeze, get a reading of fluorescence to see what nucleotide was added, then you continue until the entire sequence has been um, created. And so the entire time that this is happening, the computer is looking at your glass lane from above you. And so it's not just looking at one sequence and its fluorescence reading. Millions of sequences at once are being looked at from the above view to get all the it's like little fireworks going off, and the computer is reading all of those little fluorescent signals, and each position is unique. And the computer has a barcode for what position that is so that you can get a unique reading for that one position and get that one sequence there. And at the end, what you end up with is tons of these sequences, these fragments that have been duplicated, so that you can use that same, puzzle mapping system where there's overlap, and you make a continuous sequence based on the overlap of all your different fragments. Okay. Okay, and so there are, different types of next generation sequencing, but there were even newer, more advanced forms of sequencing that have been formed since called third generation sequencing. And so in this case, we've added some, techniques that help improve sequencing, its efficacy, its accuracy sometimes, or the size of fragments that we can sequence. Because next generation sequencing relies on small fragments, and then, there are little puzzle pieces you have to put together. Um, obviously it would be a lot easier if we could just sequence an entire chromosome as one piece without having to, do that whole mapping system. And so that's where third generation sequencing really helps. So PacBio, this is probably the most commonly used third generation sequencing platform. And there's an explanation here in text, but I'll just go ahead and move on to the next slide. Um, And yeah, I'll explain it here. So with PacBio, the way that this one works, this in this case, still working with fragments. The DNA is cut up or the into fragments, or we're using mRNA, which are fragments. Um, what we do to those fragments is we add an adapter, this sequence of DNA that can be added to the end here. And what that does is it changes this from a linear fragment of DNA to this circular fragment. This is a continuous circular fragment of DNA. Then we take this circularized DNA, and just like with our next generation sequencing, we have like a chip or a glass slide that has these really tiny microscopic wells inside of them. And that's what you're seeing here is this well. hundreds of thousands of these wells in a single slide. And at the bottom of the well, what you have is DNA polymerase that's been physically attached to the well. Add your DNA sample to your chip or glass slide, and those fragments will fall into those wells. DNA polymerase will grab onto that circular fragment, and then the DNA will pull it, the sequence of DNA, kind of like, I don't know how to explain it, but it's going to pull it through, and it's going to be sequencing that circular DNA, making copies of it. And what's useful about this PacBio mechanism is that because this is a circularized sequence of DNA, the DNA polymerase can just keep pulling it through, and it'll go around multiple times. This will spin around multiple times, and we'll get many copies of this circularized DNA, so we can get a bunch of copies. And so these bunch of copies, the reason that that's helpful is, again, DNA polymerase or attack polymerase, they make errors. And so if we make a bunch of copies of these DNA fragments, um, whichever copies are by far most abundant are the copies that are the correct sequence. Um, and so every time a nucleotide is added into the growing strand as DNA polymerase pulls this looped DNA through, um, that nucleotide's added and a fluorescent signal is emitted, just like with next generation sequencing. And we have a computer that, as DNA polymerase is doing this, it's reading the fluorescent signals in real time to give us the sequence. It's a lot easier than having to go through and map it out. Um, yeah. Okay. And so it has its advantages. One, it can do longer fragments. Two, many copies because of its circular nature of the DNA. And then we get a real-time reading. But it also has its flaws in that there are some still more precise sequencing platforms. Um, some NGS platforms are still way more have a higher accuracy, and so with some really large fragments, for example, PacBio had been known to make some sequencing errors, a lower accuracy rate. Sequencing is longer, um, and it costs much more. It is more expensive than next generation sequencing. Um, even yet another platform is nanopore sequencing. And so with nanopore sequencing, what we're doing here, and what's unique about this is this one can sequence entire chromosomes. It can sequence very long continuous strands of DNA or even RNA. And the reason for that is its mechanism is different. What we're using here instead is we have an instrument where we have this little panel or a membrane, and in that membrane we have a channel protein that's able to pull DNA through. And what we have is an electrical current induced across our membrane, meaning what we have on one side is a negative charge, and the other side we have a higher positive charge, and we use ions for that. As DNA runs through, every time a nucleotide passes through and across the membrane, we'll get a charge reaction here. We'll get a slight change in membrane potential, and we can read that with probes, yeah, electric probes. And so every time a nucleotide comes through, we get a change in current here or membrane potential. And each nucleotide will have its own unique signal. So an adenine will have a unique signal that's different from a guanine, that's different from a cytosine. And so that'll give us a reading of the entire sequence as it's just pulled through like a chain of beads. Um, this is even cheaper, but the error rate's even higher than our next generation sequencing in our PacBio. But in terms of that cheapness, just to really give you an idea, if we're looking at Drosophila, um, we've been able to sequence 15 different Drosophila species that had their entire genomes for $1,000 each. And their genomes have around 18,000 genes. So per fly, $1,000, but we're getting 18,000 genes sequenced. But Sanger sequencing, in which we usually charge, its charges $5 per sequence. If we're trying to do a fly genome in that case, 18,000 times 5 is $90,000. And so next generation sequencing has completely changed genetics in that it's a lot cheaper, and we get a lot more information out of it. Okay, so some more applications or some more techniques that we can use that are genetic techniques include DNA fingerprinting. So maybe you've heard of this, and this is where we are able to sequence human samples to compare with samples from crime scenes, whatever that might be, to see if we can find a match between a suspect and DNA found at a crime scene. And so we call this DNA fingerprinting or DNA profiling. And it works based on using those small repeat sequences that we called microsatellites. And so um, if we can also refer to our microsatellites as short tandem repeats. And what's interesting about these sequences is that between different individuals, we'll have specific regions of the genome that will have microsatellites present there. But between individuals at those loci, different individuals will have different numbers of repeats of that specific particular repeat. And so let me bring up an image so hopefully that makes more sense. Um, Here is the same loci, or sorry, same locus in different individuals, and we can see that different individuals can have different numbers of repeats. And so you might have 7 repeats in yours, where I might have 8 repeats, where Lauren might have 9, and Jacob might have 11. So every individual will have different numbers of the repeat present for that particular locus. Because of that, um, if we take sample of a crime scene and take samples of a suspect and compare their microsatellite repeats at certain loci, if we see a match, it's very likely that that suspect or that individual is a match to the DNA present at the crime scene. And so this relies on techniques that we've already talked about. So one, it relies on DNA extraction, two, PCR, three, gel electrophoresis. So DNA's got to be extracted from individual and from a crime scene. We got to amplify the DNA using PCR, specifically those microsatellite sites. And then we got to run it through gel electrophoresis to see if our sample matches the size of the crime scene. And so let me go ahead and pull open this image so we can see how that works. Again, individual one in this case, at a specific locus, they will have their unique number of repeats that individual two will not have. After we've amplified that, we can run both samples through gel electrophoresis. And what we're seeing on the gel is this individual, since they have 8 repeats, it travels a shorter distance. This individual has 2 repeats, so it travels even further. And so let's move on to the next slide real quick. When we run, yeah, actually, let me just go back. Well, if we had a separate lane here where we took sample from a crime scene and we saw that the sample from the crime scene ran to this distance here, we know that it's going to match this suspect here, individual one. And so this person is likely the person that was present at the crime scene. This can be used as this direct evidence for this individual being present. Now, while people tend to differ in number of microsatellite repeats at a particular locus, there still is, by random chance, there will be some people that have the same exact number of repeats at a specific locus. And so in that case, typically when it comes to these crime scene analysis, running this DNA fingerprinting, multiple probes, sorry, not multiple probes, multiple loci are used. The more loci you use, the more or the less likely it is that an individual will match by random chance. And so, to add on to this, a lot of times it's not even the gel that's being looked at directly. Fluorescent labels are added to different loci that are all ran at the same time through either gel electrophoresis or a capillary system, and computer can analyze the sequences as they pass through an analyzer. And what the computer can analyze or what they're getting a reading of is one, how quickly did that particular sample run through? Because the quicker it ran through, the smaller that sequence is, the more the fewer microsatellites in that position. And then two, the fluorescence reading for different microsatellite labels. And so in this case, what we have is a sample that was ran through four different loci. We can see them labeled here, these four different loci, and basically how quickly they ran through, so their relative size. Um, this one here, relative size and base pair, let's see, halfway point, maybe like a little over 100, 130, something like that. Whereas over here, it's closer to 350 size. And then what we're seeing here, this number, is the number of repeats found in that particular locus. And the reason, though, that we're seeing that these, for example, this repeat has 24 to 28 repeats present in that microsatellite. Why is this fragment smaller than this one that only has 10 repeats? And that's because those repeats can range from two nucleotides to like four or five nucleotides, which will completely change the size of that fragment. So if this one, for example, is a two nucleotide repeat, total, 24 plus 24 is 48. This one might have six repeats or six nucleotide repeat. So 6 times 10 is 60. Uh, okay, so let's go ahead and move forward. The FBI has compiled a complete list of microsatellites in humans that they can use for crime scene analysis. And so a bunch of uh, different microsatellites will be used, or these sort of tandem repeats. In this image here at the right, let me move this over again. We're seeing a suspect sample, suspect one, suspect two, and then crime scene sample. So if you're looking at this, you have to look to see which of these two suspects better matches our crime scene sample. And I think it's pretty obvious that it's suspect two here. We get a direct match there. The other thing I forgot to mention too, that I should note is that when we see a single peak like this in this reading, it's because this person's homozygous for that repeat. Both of their alleles of that locus have the exact same number of microsatellite copies. But when we see two smaller peaks, it's because they're heterozygous. One allele contains fewer copies, the other one contains two more copies in this example. Okay, so another concept check. How are microsatellites detected? I am running way over time, so I'm just going to go ahead and move past this one. Um, actually, let me pause this. There we go. Okay, so there's only a couple slides left, so let me just finish it up. Yeah, a little tired here, though, not gonna lie. Okay, so what about if we're interested in finding out what genes are associated with diseases or functions of genes, or portions of the genome? We can use two different avenues of trying to determine the gene's effect on phenotype. Um, and so one of those is what we call forward genetics, and that's where we make an observation of a phenotype. So, for example, this person has polydactyly, an extra finger. Um, we know they have the phenotype, so let's try to see what genes are associated with that phenotype. And that's called forward genetics. Make the observation of the phenotype and go from there. Um, on the other hand, we have what's called reverse genetics. And so this is the case where we have some genes where we're specifically interested in these candidate genes that we think might have an effect on this particular phenotype or this process. So let's start making mutants of that gene and see how it affects the phenotype. And we'll go forward from there. And so we call that reverse genetics. And so typically, reverse genetics relies on what we call targeted mutagenesis. We have our candidate genes, let's mutate them, and then let's see what the phenotype or phenotypic result is. Okay, and so when it comes to forward genetics, um, Random mutation is what was historically used, meaning that you would take an organism like a fly, submit X-rays to different parts of the fly, so you're changing the phenotype. After, not culturing, but after, I don't know, I cannot think, mating the flies, having them reproduce, seeing what mutations develop in offspring, or if they developed in the individual that was directly exposed to the X-ray. We see a change in the phenotype. We observe that phenotype. So maybe we see that when we submitted X-rays to the eyes, we see mutation in the eyes. Um, we go take that phenotype and try to find what genes were associated with that change in phenotype. And so that's why that's an example for genetics. We're making random mutations, but only to change phenotype. And then when we find the phenotype that we like, we'll go from there. Um, okay, I already mentioned all of that. On the other hand, if we're looking at reverse genetics, am I still recording? Okay, anyway, if we're looking at reverse genetics, let me pause this again. Oh my God. Sorry, y'all. I just realized for that last like 5 minutes, I was completely talking without screen sharing. Okay, so I'm just gonna continue on because it's getting late. Um, So when it comes to reverse genetics, this relies on targeted mutagenesis. And so the most common way today is to just insert mutations using CRISPR-Cas9 into the loci that are we're interested in. You go ahead and make single base pair changes, or you can make larger damage like double-stranded breaks and stuff, and then see what the effect is. But historically, restriction enzymes have been used in vectors. Um, and so, a really good or interesting example um, was in fruit flies, determining the function of Hox genes and kind of getting an idea of how those work. So, if you don't know Hox genes, these are body plan genes that basically determine the development of the body of an organism. So, the position of the head, the legs, arms, stuff like that, etc. And so, they were playing around these Hox genes by taking the gene for that were present where leg development's supposed to be, switching that with the loci found where antennae are supposed to grow. And so in this image, what we're seeing is normal antenna in this top, but then by changing the position of the gene for leg development and putting it where the gene is for antenna development, they found that legs grew in the place of the antenna. And the reason for this is because um, the Hox genes, the way that they're actually positioned on in the genome on chromosomes is similar to the layout of the body, meaning that genes for the head region and stuff are found at the top of the chromosome, genes for the middle bodies towards the middle, genes for the abdomen and lower below are below or lower in the chromosome. Um, and so when they started switching them around, their positions, they started noticing that, yeah, we're seeing uh, the change in growth, that these Hox genes need to be in a specific order to help proper anatomical development. Here, they made a duplication of specific genes, Hox genes, for the wing development. Um, and so they made a lot of progress in looking at this, using this targeted mutagenesis. Okay, so go through this concept check. The answer for this one, this one's reverse genetics B. And so the reason for this is because they already knew what genes they're working with, and then they made targeted mutation in those genes um, to see the result on the phenotype. Okay. Um, all right, y'all. Ugh, I'm so close. I should just finish this. Okay. And so another way to study function of DNA is to make transgenic organisms, meaning we take the gene we're interested in and we modify it and then put it into an organism to see what happens with them. So sometimes maybe we'll modify a gene that's already existing in the organism, or sometimes we can even take genes that don't exist in the organism and see how those genes affect phenotype when we do add them. And so the image on the right, what we're seeing is this mouse was uh, transferred a gene linked to obesity, a mutation that causes obesity. And so this was to help show support for obesity being linked to genetics. And so there's some different ways, but basic steps that I'll go over is the way that we can create transgenic organisms is we have two mice, male, female here, mate them so that they can undergo fertilization. And during fertilization, there's this period where the nuclei have not fused yet because it takes a couple of hours. And so we call these pronuclei because each of these are haploid instead of diploid because they haven't fused yet. And then to one of those pronuclei, you can insert genetic material for the gene of interest, and through homologous recombination, it can be added into that the chromosomes in that pronucleus. Then you'll go ahead and take this embryo, or sorry, this fertilized egg. Um, Go ahead and insert it into the mouse, implant it so it can start growing, and then she'll give birth to offspring. And then we can genotype the offspring to see which one actually has our sequence of interest. Whichever one does have the sequence of interest, we can then mate them. Um, Mate any of those individuals that have it, and then when we mate them, we can ensure that their offspring will contain the mutated version. Another thing we can do is knockout. And so knockout means that we basically remove a sequence of DNA or just completely shut it down. And so one way that can be done is um, by inserting the gene, or sorry, inserting a probe or inserting another sequence reporter gene within the target gene. And by doing so, we break it up and so it loses its function. Then we go ahead and take that genetic material, add it to embryos, take those embryos, and then we'll go ahead and through homologous recombination, those embryos will uptake the sequence. We could take the embryos or these eggs, I shouldn't say embryo, sorry, and then we'll go ahead and add it to a growing mouse embryo. That's a genetic match. And then when we add the embryos in, these will go ahead and grow into an organism that will have cells with that genotype, and then we'll have them mate. And then once they mate, their offspring should contain it. That is the end of the lecture. So here's some takeaways. If you have any questions, just email me, let me know. The homework, since I am getting it to you kind of late, I'm going to make it due next Tuesday instead of Sunday. So there's some extension for that. Um, and then the quiz as well, Tuesday next week. Hopefully, y'all had fun at Little Verna Palooza. Hopefully, you have a good night. Um, and yeah, if you have any questions, just let me know. Bye, y'all. Okay, not bye yet because I need to stop this.